Have you ever wondered how your brain adapts and learns new things? This remarkable agility is due to a phenomenon known as neuroplasticity. Neuroplasticity is the central nervous system's inherent ability to dynamically adapt and reconfigure its structural and functional architecture. It's like the brain's own personal renovation team, constantly redesigning and reshaping to ensure everything operates at its best. Neuroplasticity isn't limited to just one area of the brain. Rather, it's a global phenomenon encompassing every single neural pathway and synapse. It's a response to an intricate web of both internal and external stimuli. Think of it as the brain's way of adjusting the knobs and dials to fine-tune its performance based on the demands of our environment and experiences. Now, you might be wondering, how does this happen? Well, it's a complex process involving several different mechanisms. One such mechanism is synaptic plasticity, a focal point within neuroplasticity. Synaptic plasticity orchestrates a mesmerizing molecular flux at the synaptic level. It involves an intricate interplay of neurotransmitters, which serve as molecular messengers interacting with their cognate receptors on the postsynaptic membrane. This dynamic dance of neurotransmitters, such as glutamate and GABA during synaptic transmission, contributes to the delicate modulation of synaptic strength. It's a molecular ballet, if you will, with excitatory and inhibitory inputs, shaping the postsynaptic response. But it doesn't stop there. Synaptic plasticity is further shaped by the dynamics of neurotransmitter receptors, such as the NMDA and AMPA receptors in glutamatergic synapses. These receptors undergo structural changes and alterations in their conductance, influencing synaptic transmission and participating in the induction and expression of long-term potentiation and depression. Beyond the synaptic cleft, intracellular signaling cascades play a pivotal role in translating extracellular signals into enduring changes in synaptic efficacy. It's a biochemical symphony that orchestrates the events that govern synaptic plasticity. Intriguing, isn't it? How our brain constantly reshapes itself. This is just the tip of the iceberg when it comes to the enigma of neuroplasticity. In the next few minutes, we'll delve deeper into this fascinating subject, exploring the molecular ballet that is synaptic plasticity, the role of neurotransmitter dynamics, receptor dynamics, intracellular signaling cascades, and much more. So stay tuned as we unravel the mysteries of the brain. But how does this happen at the molecular level? Let's delve into the dance of synaptic plasticity. This captivating ballet unfolds at the synapse, the junction between two neurons. The neurotransmitters, our molecular messengers, take center stage. They glide and pirouette from the presynaptic neuron across the synaptic cleft to the postsynaptic neuron, where they interact with their respective receptors. Picture glutamate, an excitatory neurotransmitter, and GABA, an inhibitory one, as two lead dancers, each performing their unique roles. They modulate synaptic strength, the intensity of the neural connection, through their dynamic ebb and flow. Then there are the receptors, the audience of this molecular performance. In glutamatergic synapses, for instance, NMDA and AMPA receptors attentively watch the performance, changing their structure and conductance in response. Their reactions influence synaptic transmission, shaping the postsynaptic response. This molecular dance, with its complex interplay of neurotransmitters and receptors, forms the foundation of synaptic plasticity. Now imagine a dynamic ebb and flow of neurotransmitters. These neurotransmitters, like glutamate and GABA, are the molecular messengers of the brain, transmitting signals across the synaptic gap. Picture them in an intricate dance, moving rhythmically between neurons, their movements shaping and reshaping the synaptic strength. This dance is not random, it's precise, it's calculated, it's necessary. In the grand ballet of neuroplasticity, neurotransmitters are the dancers, but they don't perform alone. They interact with their partners, the neurotransmitter receptors, such as the NMDA and AMPA receptors in glutamatergic synapses. These receptors are like choreographers, guiding the neurotransmitters, responding to their movements and altering their own structure and conductance in response. It's a continuous feedback loop, a dance of mutual influence. This dynamic interplay between neurotransmitters and receptors forms the foundation of synaptic plasticity. It's like a molecular seesaw with excitatory and inhibitory inputs shaping the postsynaptic response. This delicate balance is critical, as it modulates synaptic strength, influencing how signals are transmitted and processed within the neural network. But why is this dance so important, you may ask? Well, these dynamics are what make your learning and memory possible. They are the mechanics behind the brain's ability to adapt and reconfigure its structural and functional architecture in response to various stimuli. The changes and alterations in the dance of neurotransmitters and receptors facilitate the induction and expression of long-term potentiation and depression. Long-term potentiation is akin to the dancers rehearsing a particular move repeatedly, strengthening the synaptic connection, enhancing signal transmission, and fostering learning and memory. In contrast, long-term depression is like the dancers taking a rest, reducing synaptic strength, and allowing for the elimination of unnecessary or redundant connections. So next time you learn something new or recall a cherished memory, remember the intricate dance of neurotransmitters and receptors that made it possible. These changes and alterations are what make your learning and memory possible. Beyond the synaptic cleft, there's an intricate signaling cascade. Picture this, a relay race where the baton passes from one runner to another, except here, the runners are molecules. This race is pivotal in translating extracellular signals into enduring changes in synaptic efficacy. Intracellular signaling cascades, as they are known, involve second messengers, kinases, and phosphatases. These aren't just any runners, they're seasoned athletes, each with a specific role in the race. Second messengers like cyclic AMP are the first to receive the baton, passing the signal from the cell's surface to its interior. Kinases, the next runners, are protein enzymes that add phosphate groups to other proteins, altering their function. Meanwhile, phosphatases are the referees, removing phosphate groups and ensuring balance. This relay race is far from a simple dash. It's a marathon, a complex and coordinated series of events that ultimately govern synaptic plasticity. These cascades coordinate the biochemical events that govern synaptic plasticity. Synaptic plasticity is often exemplified by long-term potentiation and depression. In our exploration of the brain's adaptability, we reach the fascinating phenomena of long-term potentiation, or LTP, and long-term depression, or LTD. These are mechanisms by which the brain strengthens or weakens connections between neurons. Picture it as a friendly conversation between neurons where one neuron says to the other, "I find our chat so stimulating, let's talk more often." This is achieved through an increase in the release of neurotransmitters or a rise in the number of receptors on the receiving neuron. This amplifies the signal, making the connection stronger and more likely to be activated in the future. Now imagine LTD as the opposite, a gentle way for neurons to say, "Our discussions are less exciting than they used to be. Let's chat less." This happens when the release of neurotransmitters decreases or when the postsynaptic neuron becomes less responsive, scaling down the signal strength. The connection weakens, making it less likely to be activated. These processes are not just about quantity but also quality. The molecular events that occur during LTP and LTD are intricate and complex. For instance, in LTP, there might be an increase in the trafficking of AMPA receptors to the postsynaptic membrane, enhancing its sensitivity to glutamate, the main excitatory neurotransmitter. On the other hand, LTD might involve the removal or internalization of these receptors, making the postsynaptic neuron less responsive to glutamate. It's like adjusting the volume of a conversation, turning it up during LTP and down during LTD. Moreover, these alterations are not just transient. They may trigger downstream signaling cascades, leading to changes in gene expression and protein synthesis. This can result in the growth of new synaptic connections or the retraction of existing ones, permanently altering the neural circuit's architecture. These phenomena entail complex molecular events, including changes in receptor trafficking, synaptic protein synthesis, and alterations in gene expression. Synaptic plasticity operates within the broader context of metaplasticity. This is a concept where the past activity of a synapse influences its future ability to change. Imagine it as a form of plasticity behind the plasticity, a second layer of adaptability that fine-tunes the synaptic changes based on their prior history. Now, while metaplasticity ensures the synapse's adaptability, there's another key player maintaining the stability of neural networks: homeostatic regulation. This mechanism acts like a neural thermostat, adjusting the synaptic strength to maintain an optimal activity range. It's a balancing act that ensures our brain doesn't go into overdrive with too much excitation or fall into a lull with excessive inhibition. These two elements, metaplasticity and homeostatic regulation, work hand in hand. They underpin the incredible adaptability and stability of our neural networks, enabling our brains to learn, adapt, and respond to the changing world around us. This is how our brain maintains an optimal range, preventing runaway excitation or inhibition. Neuroplasticity underscores the brain's innate capability to adapt, optimize, and restructure. This remarkable feature is the lifeblood of cognitive flexibility, allowing our minds to adjust and evolve in response to the ever-changing complexities of our internal and external environments. Picture a symphony orchestra, where each instrument represents a different component of our neural network. Just as a conductor guides the orchestra to adapt to the fluctuating tempo, rhythm, and dynamics of a performance, neuroplasticity coordinates the various elements of our brain to optimize its function and adapt to new circumstances. This is the epitome of cognitive flexibility. From the synaptic ballet of neurotransmitters and receptors, to the intricate choreography of intracellular signaling cascades, these processes all play their part in shaping our brain's adaptability. Neuroplasticity, in essence, is the testament to the cognitive flexibility inherent in our brains.